Biotechnology and its application, molecular diagnosis part 2. In the previous video we have already told you what is the essentiality or importance of diagnosing any disease as soon as possible it is. Early diagnosis was very much important for the effective treatment of a disease, and we had also told you that in this aspect biotechnology will help us. In conventional method it was not possible to detect any pathogen at its early stage, but biotechnology has developed many techniques which will help us in early diagnosis of any disease. And we had also told you that two most familiar techniques, one is PCR and one is ELISA. In this topic we will discuss how PCR means polymerase chain reaction, helps us in early diagnosis. So let us discuss about PCR. First, remember what is the actual function of PCR, what PCR does actually. PCR actually one amplification machine, PCR actually amplifies nucleic acid contents. PCR, it actually amplifier. It makes clones actually. And in most cases, PCR is used to make the clones of nucleic acids, clones of nucleic acids, DNA or RNA. If you give a small piece of DNA or RNA into the PCR system, then PCR will multiply it into billion times within some times. So the main role of PCR is amplification of nucleic acids. So let us now see how PCR will help us in early detection or early diagnosis of a disease. Nowadays, this PCR is being routinely used. You probably have heard the word HIV. You all know about the disease. You know about the fatal disease cancer, and you are also familiar with a lot of genetic disorders. You also know about mutations. So PCR will help us in detecting those all diseases. How? Let us see. So first let us see in which cases PCR is mostly used. The use of PCR is very famous in HIV virus detection in suspected AIDS patient. Next, PCR is also used in case of cancer. Cancer may be caused by a lot of means, by various means. One of the main reason behind cancer may be mutation. Mutation in the gene may cause cancer. In that particular cases, this PCR system or this PCR technique will help us in detecting any mutation in suspected cancerous patient too. So let us write down now detecting mutation in gene in suspected cancer patient. So in case of cancer, we will use the PCR technique to detect mutation. Actually, PCR will detect mutation. So mutation is related, any other disease can be detected by PCR. A lot of genetic disorders is familiar within our human community. Those all mutation related diseases or genetic disorders can be detected by PCR. Third, we will write that many other genetic disorders will be also detected using PCR. So here we are using one word, HIV detection, mutation detection, or in any genetic disorders we are using the word detection. But if you remember the procedure of PCR, then you will not find anything in the PCR where PCR detects anything. What PCR does actually? PCR actually amplifies. It makes clones of nucleic acids. So it is very simple to understand that PCR alone cannot detect this all. PCR will help obviously. PCR, what will do PCR? Suppose there is a very little amount of HIV virus in your serum or in your body. You take the sample, you multiply the copies of that particular HIV virus using the PCR. Suppose there is a small gene, you are suspecting that there could be some mutation. So then you take out the gene, you give it into the PCR system, PCR will amplify it, PCR will make millions and billions of copies of it. In the same way, if you suspect any genetic disorder, you give that particular nucleic acid piece into the PCR system, PCR will make its clone, and PCR will make a lot of copies of it. So PCR alone cannot detect. PCR will help in only making clones of that particular molecule, that may be nucleic acid, that may be any virus particles, nucleic acid, anything. So for detecting, we have to take the help of probe. We have to take the help of probe. Let us discuss what is probe actually. We have to take the help of probe. So let us discuss what is probe. What is probe? Probe may be created with any molecule, but in this chapter and particularly in this topic, we will discuss the probe with respect to nucleic acids only. So probe is a single stranded, remember this point, single stranded nucleic acid. Single stranded nucleic acid. It must be single stranded. Probe never generated in a double stranded nucleic acid. Probe always generated or always produced in a single stranded nucleic acid. So nucleic acid may be two types. One is DNA and one is RNA. So RNA is normally single stranded, there is no problem. But you know DNA is double stranded. Then what you have to do? This DNA, you have to denature it. Denaturation, if you do the denaturation, so then ssDNA strand can be obtained. So this in the single stranded DNA and RNA, how we will make probe? Let us see if you can remember the structure of the nucleotide chain. To understand this probe, we must remember the molecular structure of the nucleic acid. Let us simply draw one sketch of the nucleic acid. So this is one pentose sugar. This is one pentose sugar. This is another one pentose sugar. So what was there? This is pentose sugar. This is one carbon, 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 carbon. There was also one carbon, C5. This is C1. There is carbon, 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 carbon. There is also one carbon. Okay, this is oxygen. Carbon, 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 carbon. There is also one carbon. These are pentose sugar actually. These are pentose sugar. And you know, with that number one carbon, nitrogen base is present. Any nitrogen base is present. There is nitrogen base is present. And this structure you all know. Pentose sugar plus nitrogen base is called nucleoside. Now these structures will be added by disulfide bonds. And where the bonds will occur? It will be occur within this prime carbons, there will be a phosphate group. There will be a disulfide bond. After knowing this structure, after analyzing this structure, we can conclude that the main atom which are present in a nucleic acid, that is carbon, nitrogen, phosphorus, these are hydrogen, these are some important atoms which are present in the nucleic acids. So now to make probe, what we have to do? If we just take one single stranded DNA, this is not a probe. In the next stage, we have to label it, or we have to tag it with radioactivity, or nowadays fluorescent properties also used, but here we will discuss only about radioactivity. So here tagging. Next stage is tagging or labeling. With which radioactive tagging we will do. Radioactive tagging. And how this is occurred? Actually, when we produce probe, we artificially synthesize the nucleotide sequences. We artificially synthesize this DNA or RNA sequence. And during that time, we use any of the radioactive isotope of these important atoms. And the mostly used is P32. Phosphorus radioactive atom is mostly used. So remember, if this is P32, this is P32, that means in this P-shaped DNA or RNA nucleic acid, there is one atom which is radioactive in nature. So this fragment or this part of nucleic acid, which is single stranded, which is tagged or labeled by any radioactive molecule, any radioactive atom, this structure will be now called a probe. This is now a probe. Now let us see how this probe and PCR will work together and will help us in detecting even a minute amount of HIV virus, or it will help us in detecting if there is any mutation in our body. Let us see this. Let us now discuss how this PCR and DNA hybridization and probe will help us in detecting mutation in any gene. Suppose this is a piece of gene, this is one gene or DNA fragment, and you were suspecting that there could be a mutation. So first you take it, you use PCR on it, make many copies. Okay, make many copies, and also you have to create the complementary probe against this. For example, you know the sequence of the gene which you are suspecting. If the gene is normal, its nucleotide sequence may be like this: ATGC, ATGC, ATGC. Its nucleotide sequence like this. It may be ATGC, ATGC. It may be ATGC, 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 ATGC. This is the normal sequence. Okay. In respect to this, you have made one probe, just complementary to it. TACG, TACG. You have made probe TACG, TACG, and this probe is probes are radio elaborate, you all know. So now this probe is added to these clones of cells, to these clones of nucleic acids. And for example, say that in this particular segment of gene, there has been a mutation. There has been a mutation. There is C and there is A. It is the mutated form. So now what will happen? This probe will come. This probe will attach with this as this is complementary. Will attach also with this as this is complementary. Will attach also with this as this is complementary. It will attach also with this as this is complementary. But this probe will fail to attach with this because there some mutation has been occurred. And as the mutation has been occurred, so nucleotide base pairs has been changed. So probe will not be complementary here, and they will not bind. There will be no hybridization of the DNAs. Now the simple procedure we use autoradiography. We'll use autoradiography. There will be a photographic film. We will get it from here. We'll also get it from here. We'll also get it from here. We'll also get it from here. But from here, nothing can be obtained. So in this way, where seeing the photographic film using the radiography, from which segment of the gene or from which segment of that particular nucleic acids, from where there will be no radiography, there will be no radioactivity, we can simply say that there is a mutation because it was complementary to it. As the mutation has been occurred, so complementarity has been lost and it will not be added to this, it will not join to this, and there will be no radioactivity. In this way